Okay, so let's get started. Yep, people have not joined yet, but we'll get started. Okay, okay. Now, welcome to week 6 of Next Generation Sequencing and Technologies, Data Analysis and Applications. So, this week we'll be mainly talking about variant calling. What are the different types of variants and what are the different algorithms and softwares used for variant calling? And we'll also see the format in which the variants are stored, whether it is a VCF format, how it looks like, and what are the different caveats for it. Okay, yeah. So this week we'll mainly look into variants and how to store the variants in different formats, basically VCF, and how to run downstream analysis on those as well. So annotation and functional impact and stuff like that. And before that, we'll uh, touch up a little bit about different types of yeah. We'll touch up a little bit about different types of methods that we use to pre-process reads after alignment. So we talked about alignment and its pre-processing in the previous classes. We'll talk about post-alignment processing and different types of pre-processing that is done on that, and after that variant calling, and finally variant annotation. Okay, so let's get started. So first question comes to mind is, why are genomic variants important? Why do we want to find variants from after doing WGS on a particular sample? Why do we want to find variants? Because in most of the papers on research projects, you'll see after WGS sequencing, the thing that they do is ultimately variant calling, and they study the variants. Now the question comes, why is it important? So people's genome are far more similar to each other than they are different. It is frequently stated that any two people's genome are almost 99.9% identical to each other. Like, there are different caveats to it. If people are from the same ethnicity, same ancestral background, they'll have more similar variants. People having different kind of ancestral backgrounds will have different kinds of variants, but on an average, it's more than 99% equal. And this percentage is based on finding that on an average, a single nucleotide difference exists between two people's genome once in every 1300 nucleotides or so. So that is a frequency, and that is how it is calculated that number 99.9 comes out. But this is not like a fixed number. This number varies between different individuals depending on primarily their ancestral backgrounds. But yeah, uh, but this is an oversimplification because it only accounts for single nucleotide differences. In reality, any two people's genome are on an average 99.6% identical and about 0.4% different. The latter percentage reflects both single nucleotide difference and differences that involve multiple nucleotides. So we'll discuss these little later, which are known as structural variants, which are not like single nucleotide changes, but a sequence of nucleotide changes that can be different types of changes. We'll talk about those a little later. The differences between human genomes are called genomic variants. A person's set of genomic variants is part of what makes them unique. Other factors, which are environmental factors such as diet, lifestyle, social context, also contribute to a person's uniqueness. Uh, but most variants have no influence on the functioning of a person's genome, but a small subset of variants do have an impact. So, yeah, now a small subset of uh, these variants do have an impact, and these are the variants that cause diseases, that cause different types of phenotype changes in a person. Say, for example, a person is more susceptible to a high blood pressure. That might be a reason because there are certain variants that are associated with high blood pressure, are present in that particular individual. Uh, yeah. So For example, some genomic variants influence physical characteristics such as eye color, hair color, height. Uh, Others influence health conditions or how a person responds to certain medications. So this is also very important in terms of pharmacogenomics. Is a drug will not be as effective, as equally effective to everybody, because the genome of every individual will not be the same. There will be variants in particular individuals which will contribute to drug resistance. There will be variants that will be susceptible to the drug. So there can be different varieties of these variations that can influence different aspects of health, disease, and even medications. So, what is variant calling in terms of NGS data? So, majorly three steps in variant calling, and that includes the whole pipeline, and I have just constricted it into three steps. But it is a process by which we identify variants from sequence data. So, uh, what we do is we carry out whole genome sequencing or whole exome sequencing, and we get using different types of technologies we discussed before, Illumina short read, or it can be packed by Oxford Nanopore long read, or different types of array data as well. So, but in this case, we'll talk about mostly whole genome data. So, whole genome or whole exome sequencing data, and we get the reads in the form of FASTQ files. After getting the FASTQ files, we align this to a reference genome, creating BAM files or CRAN files, and we identify. This is the step where we identify the variants. Is basically where we identify where the aligned reads differ from the reference genome, and we write these variants into a file format known as the VC file. So, for example, these are these are two parts of a genome, basically where different. So this is the reference genome on the top. The colored one is the reference genome, and the gray sequences that you see, these are different reads that are aligned to the reference genome. So we have, say for example, this read. So in this read, in the first read, as you can see, is a very long read. It covers almost the whole part of the genome. But at this point, you have a conflicting nucleotide here. So in the reference, there is an A, but in your read, there is a C. But in as you see, there in other reads supporting in the same location, there's an A. So what can happen is what you'll have is during alignment you'll have multiple reads aligning to the similar position. So one position will be covered by multiple reads, as we discussed in coverage and sequencing depth and stuff. So a particular region of the genome will be covered by multiple reads, and each of those reads will tell you what the nucleotide is in that position. So, what we do is we take a consensus. So, for example, in this case, only one read is telling you there is a C in this place, but the rest of the reads are telling you there is an A. So, it's most probably that there is an A here uh, in your sample read as well, and this is just an error or a sequencing error or some sort of error that has been introduced into this field. So we look at multiple evidences of this change in nucleotide in the sample compared to the reference. So and that is how we find different types of variants. Now this will be, say for example, this will is not actually a variant because there's only one uh, read supporting this particular variant. The other reads are not supporting. But here there are enough evidences to claim that there is not a particular. Just a second. Yeah, there are many evidences to claim that there is not a single nucleotide polymorphism here. A single base nucleotide change is not occurred here. But on the other hand, if you see in this case, there is only one read supporting this particular change in nucleotide, which is T2A compared to the reference. Now, this this one, if you claim to be a single nucleotide polymorphism or an SNP, this is a little bit dangerous because you do not have enough evidence. You do not have enough evidence to claim that this is a SNP. So, depending on how many reads support and what kind of nucleotide changes occurred, will all have a quality score associated with it, and also a depth associated with it. So in the VC file, we'll see later there will be a depth parameter which tells you how many reads are supporting this particular variation. So depending on that, we filter our variance and we uh, only take the high quality variants and do further analysis. So this is just to give you an overview how we do, how actually you can look at a actually you can visualize what are SNPs in the context of alignment and read depth. Okay, so these SNPs or SNVs, what we call them, are there are various types of these SNVs. Uh, the smallest variants are known as single nucleotide variants. Each SNV reflects difference in a single nucleotide. For a given SNV, a DNA letter at the genomic position might be a C in one person, but a T in another person. So basically, this example. So this is so this is one matter. This is a person which has this maternal and paternal chromosomes, and this is person 2 and person 3. In some persons there will be a C to T change. In some person it will be the C in both strands. In some it will be a T. So this type of variation compared to a reference, so we always uh, define SNPs or SNVs, single nucleotide variants, in context of a reference genome. So, and one thing to remember is this reference genome is a standardized genome, but it does not. It is a standardized European genome. It's not a Indian genome or an African genome or any other like a genome from any other continent of the world. So it is specifically an European genome. So what happens is so what happens is basically, even though if say for example you have taken a reference genome for a human and which is like basically mostly European genome. So if you take a sample of Indian origin or of African origin and sequence the sample and you try to align it with the reference genome which is obviously European, you'll find a lot more variations compared to a Europe if you align a European sample to that particular genome. So the reference is always you have to remember that the reference is not always perfect as well. So these variants, what you see, they might be a variant in context of a reference, but if you sequence 10 more individuals of the same ethnicity or the same ancestry, you'll find that all 10 of them have the same variation. So in the context of that particular ancestry or the or basically the region of the genome of those particular individuals who are like who have a separate ancestry compared to the reference, uh, those are not variants in context of their genome. So if you made a reference genome based on African sample, then those variants will not come up because most of the African people will have the similar kind of SNP structure. So the variants are always defined in terms of the reference genome, and you have to take it with a pinch of salt. But the reference genome is always uh, and like or not always, but currently the reference genomes that we have are mostly European based reference genomes. So the Indian samples and African samples and samples from other parts of the world will have much more SNPs compared to a European sample because we have compared it against a European reference. So just keep this in mind. So to be considered a SNP, a single nucleotide variant must be present in at least 1% of the population. In terms of the population, we do not have access to sequence data for the whole population. So, in this case, the population meaning the cohort of the samples that you are trying to sequence. So, there are cohort studies like the 1000 genome or the Nomad, or there are other studies as well where they sequence thousands of individuals where they sequence thousands of individuals and they check for different SNPs and SNVs. So this 1% is in context of the cohort of the samples, not exactly the population, because you do not have the data for like what are the variants for each individual of a particular population. So you'll have a subset of that, and in terms of that only, you consider at least 1%. So SNV is a much more general term, which includes both relatively common, which is the SNP, and the rare nucleotide differences as well. So SNP Single nucleotide polymorphism is a term that is mostly referred to common nucleotide changes, but sorry, SNV is a term that is commonly used for nucleotide changes, where such as SNP uh, is like if that uh, variation or the nucleotide change is present in at least 1% of your population. So yeah, and another group of so we talked about single nucleotide variants. So a single nucleotide change. So C to T or a C to G or a C to A, whatever we there is a single nucleotide change. A single nucleotide has been changed, but there are other types of genomic variants as well, which includes the small insertions and deletions inside the genome. So these are often known as indels together. Insertion or deletion variants reflect an extra or a missing DNA nucleotide in the genome, respectively, and typically involve fewer than 50 nucleotides. This is important because the moment you go above 50 nucleotides, we have a separate term for it, which is known as structural variation. So anything below 50 nucleotide, we call it an indel, just small indels. If we go above 50 nucleotide, that's an insertion structural variant or a deletion structural variant, because those are like 50 base pair is considered to be a quite a big uh, change inside the gene. So we handle those separately. We handle variations which are less than 50 base pairs separately. So, okay. So insertion deletion variants are less frequent than SNVs, but can sometimes have a larger impact on health and diseases by disrupting the function of a gene that encodes for an important protein. So this is also intuitive. Ki, a major change in the genome will be less prevalent compared to a minor change because of evolutionary constraints. So if you have, say for example, a person is inheriting a particular indel from its parents, from his or her parents, and that indel might be deleterious in nature, might affect some sort of protein synthesis downstream, or that variant might cause a disease as well. Now, if that variant is mostly these big variants will be disruptive in nature, even if they are not disease causing, they'll disrupt the genome in some way or form, uh, disrupting protein synthesis or mRNA synthesis or whatever. So these are not evolutionary acceptable. So the person who gets this, so the person who will inherit this particular long variation has a lower chance of propagating that variation to the next generation, because person might not be alive till that age to be to reproduce and propagate it into the next generation. So this is also evolutionary intuitive. Ki single nucleotide variants are much more tolerable than a little bit longer base pair changes, which that might be like some sort of insertion deletion. Sometimes, at least most of the times, these variations are benign in nature. So although there is variation in the in terms of protein synthesis or mRNA or any kind of disease phenotype, those are silent in nature. So they do not contribute to those in these things in much. So if uh, so the most indels or most variations that you find will be of these kind of nature. A few you will find are associated with rare diseases. So because those are disrupting the gene or function of the protein function somehow. So these are some things that you need to like think about really, whether SNV is more desirable, or SV is more desirable, or whatever it is. So you have to think it from an evolutionary perspective. Feel like if the variation is too large, it uh, might not help in evolution, or the evolution might not accept. It will not be the fittest, so it will not survive. Another common type of insertion deletion variant are what are known as tandem repeats. So these are also called microsatellites. So what are these? These are short stretches of nucleotide which are represented multiple times and are highly variable among people. So these are small set of repeats. Like for example, here it is given CAG. It's a repeat of CAG, 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 something like that. So these are short stretches of nucleotides that are repeated multiple times, and these are very highly variable among people. Different chromosomes can vary in the number of times such short nucleotide stretches are repeated. excuse me, ranging from a few hundreds of times. Historically, tandem repeats have been used for building maps of the human genome and DNA profiling in forensic applications. So, these tandem repeats are actually kind of you can consider those as DNA fingerprints, which you can which is basically used in different types of applications like forensics. So this is a insertion. So a GAT has been inserted into the strand. An SNV here is just a single nucleotide change. A deletion is just cutting off that portion of the genome and joining the ends. And the tandem repeats are small stretches of DNA which are repeated one after another, like that. So these are different types of small variations that we have. Now, how are these variations inherited? That is also an important aspect of studying these variants. So there can be two types of mutations. One is the germline, one is the somatic. From the name, it's clear. So germline are mostly mutation which in the reproductive cells of the person. So that germline mutation is basically propagated to the next generation. On the other hand, somatic mutations are not present in the germ cells, which are the reproductive cells. So those are not propagated to the next generation. Those are like constricted to the individual itself. But if your germline is mutated, uh, some mutation is present in your germline, it is highly probable that variation might be propagated to the next generation. So yeah, in germline mutations, the mutation is inherited in the whole embryo, and as a result of this, this mutation is inherited in all tissues of the grown human being, because all the cells will be originating from the zygote, which will come from the sperm and the gamete. And if either of your sperm and gamete is mutated in somehow, that mutation will be propagated into all the cells of your body. And uh, if the mutation is inherited, it will be inherited in all tissues, and half of the gametes will carry these mutations, because it's if you remember, the DNA is two-stranded. So if one strand contains that mutation, there's a 50% probability that that mutation might be propagated in the next generation. In case of somatic mutations, these mutations are not present in germ cells. In early development, mutation, it can happen that a mutation is appearing in a somatic tissue. It is possible during the development, development of the embryo, some sort of mutation has taken place in a particular area of the embryo. So when that person grows up, the mutation will be there, but it will continue only in a somatic tissue, and the mutation will not be carried on into the germ cell, and it will not be propagated into the next generation. Similar is the logic for mutations that are acquired through environmental changes. Say, for example, we know that UV light mutates, uh, basically damages the DNA and mutates a part of your DNA, particularly in skin tissue. But that mutation is a somatic mutation, because that mutation is not propagated into your germ cell, so it is likely not to be propagated into your next generation. So diseases like cancer are not like cancer is a multifactorial disease, so it's hard to claim anything about cancer. But most types of cancer that we see are due to somatic mutations in particular tissues. So, for example, if a particular gene, say for example, a BRCA gene is mutated, there's highly likely that that person will develop breast cancer. But it is highly unlikely that that BRCA mutation will be propagated into the next generation, because that mutation has only taken place around uh, in the breast cells of the person, not in the germline of the person. So it's most likely not to be propagated. So most cancers are like that, but obviously there is research still going on. We do not know 100% about those, but yeah, this is a good example to see. So these are different types of mutations and variations, and like how we inherit or propagate them to the next generation. But there are certain things when you are detecting this variants using computational algorithms or bioinformatics procedures. there are some things to consider while detecting these variants. So before engaging in a project using sequencing, it is always a good idea to consider different options and choose the one that matches your research question and provides the most cost-effective solution. So this DNA isolation and fragmentation, you depending on your type of sample. Say, for example, you know that a particular sample is a diseased sample, and you have an idea key around this gene, a particular gene has been affected, and around this gene, you are expecting some sort of mutation, some sort of variations in the gene. So when you're doing this isolation and fragmentation of the DNA, you have to be careful so that you capture the whole stretch of the particular gene or the area of the genome that has the particular mutation that you're looking for. So uh, they have this DNA isolation can have systematic effects on the representation of the different genomic regions and as a consequence on variant detection. So you need to be careful if you're doing some sort of targeted sequencing or you know that you are expecting this kind of a variation in this part of the genome, then you need to be careful that you do the isolation and the fragmentation in such a way that the that the reads that you have enough evidences for a particular variation in that particular region. So you can obviously do that depending on like how you fragment the DNA. The second thing that we see that affects uh, variant detection is PCR duplicates. We'll see in the next slides as well. So PCR duplicates are basically when you are uh, creating this library of reads from the sample, you're fragmenting the DNA, isolating the DNA, and then what you do is basically amplify uh, the DNA so that you have enough reads to get matched. So this PCR amplification is done during library preparation. So what happens is like if you do not remove the PCR duplicates inside from your reads and you align those reads directly to the reference, so it can be possible that if one of your read has been detected in a wrong manner or there has been a sequencing error in one particular read, so it is highly likely that that basically variation or that particular sequencing error will be amplified in the next reads as well. So you'll have a set of reads if you do not remove the duplicates. So you'll have a same read that is present multiple times. But if you look at in an objective manner, you'll see uh, if there is a variant, that variant is supported by so many reads. But ultimately, it's actually not so many reads. It's actually one read only, which has been amplified many times by PCR, and that leads to the duplication of the reads. So you have to be a little bit careful when you're detecting variants. You do not take the evidence from one read that has been amplified. You would want multiple reads that are there, multiple different reads that are supporting that particular variant information. So removing PCR duplicates is a good idea to do before variant calling, because variant calling is based on the evidences of the number of reads supporting that particular variant. So if you're supplementing your data with repeats of the same read that are supporting the same variant, it would not be a good case. So during PCR duplication removal, what they check is basically reads having the same start and the end position that are aligning to the same start and the input position, same length of read, and you have a number of reads of that kind. So those are most likely PCR duplicates. Right? Yeah, the third thing that comes into play is the genome coverage, as I was talking about. So the coverage of the genome has to be enough so that when you find a variant, you can claim with confidence that variant is actually present. It's not sequencing error. It's not some basically a false positive or anything. So you need to have enough evidences for that particular variant. So one way you can increase the evidences is basically increasing the coverage of the depth of the sequencing. So if you have more reads actually supporting that particular variant, the better. So increasing your coverage is a good idea if you want to have a good variant detection good evidences of detection. The fourth and the final point that comes into play is the read length and the platform eventually. So, as we know, Illumina gives around 150 base pairs of read length. So if you have two opposing reads, read one and read two, which are basically your paired reads. So including read one and read two, you get around 300 base pairs. And if there's an insert size, it's slightly above that. Maximum, it's around that 500 to 600 range, considering the read one, read two, and insert. Now, this is okay for if you have single nucleotides changes. It's okay to detect single nucleotide changes with a 300 or more base pair length of read. But say, for example, if you are detecting a structural variant, you are trying to detect a repeat region or some sort of tandem repeat, as I was speaking about. So, say that tandem repeats can go on for like thousands of base pairs. Now, 150 base pair of read length is not enough evidence to claim repeat or a particularly bigger variation. So if you have 1,000, say for example, you have 1,000 base pair deletion in the genome. So it's very hard to have evidences for the 1,000 base pair deletion with reads of 150 base pairs or 300 base pairs aligning against each other. So it's there. There will be obviously evidences, but calling big reads, big variations with small reads, it's not a good idea, and it's not well accepted even. So if you have bigger reads, the better idea to go for is long read sequencing. Sequences like ONT or PacBio, you get bigger base pair, bigger read length. You can go up to like 1 KB even, is like 10 KB even. Sorry. So the the longer the read, the better evidence you'll get for bigger variations in the genome, be it insertion, be it deletion, be it some sort of duplication, some sort of repeat, or anything. The bigger the read, the better evidence you have for these big variations. But in case of smaller variations, it's okay to have small base pair length because it's just one nucleotide change, so it's easily identifiable if you even if you have smaller reads, because it's okay to support a single nucleotide base pair change with the help of a 300 base pair read, multiple 300 base pair read. So it's it is like okay to do that, but it's not very okay to claim structural variations, big structural variations. Small structural variations are still okay. 100 base pair, 200 base pairs, even 300, 400 base pairs is still okay. But beyond 1,000, it's not a very good idea to use a short read data. So these are the different types of variations and the different types of caveats that we need to consider while calling and detecting variants. So this kind of what to say summarizes the whole the last 3 4 slides in one picture. So this is your reference genome, this is your target genome. So these are your PCR duplicates, so they have the same read length and they're giving the same obviously the same nucleotide at that position. So these again are not PCR duplicates, though they are giving similar reads in similar position, but since the starting and ending are not the same, these are not actually PCR duplicates. So one thing here is basically if you see the difference between a short read and a long read. For short read, you need a very good depth or depth of coverage to actually claim a structural variant in any position. But in long read, since you have a bigger stretch of DNA that is aligning to a particular region, the number of evidences of such required is actually less. So this is the major difference between the short read and the long read in terms of variant detection. Now, one thing to notice here as well, the coverage is not uniform as well. So there will be low coverage areas like these, and there will be high coverage areas like these. So a variant falling in this region will have less confidence than a variant falling in this region, because you'll have more evidences in this case and less evidences in this case. So the thing to do is basically have high quality DNA. You have uniform fragmentation, sufficient DNA quantity, a good sequencing platform. If you are looking for single nucleotide changes, it's okay to use short read, but if you're looking for very big uh, variations in the genome, it's good to have long read data. But the bottleneck comes in our cases as well when we do these studies is basically the cost. The cost is too high for long read. It's almost like 5 to 10 times of a short read sequencing technology. So the cost is a very big bottleneck in terms of long read. But in terms of like the evolution of the sequencing technology, so if you remember, like we started with very expensive sequencing technologies in the early 2000s, and that has gone down massively if you compare costs of sequencing a sample now versus 20-25 years ago. So hopefully this will go down as well. So in the future we can all rely on long reads for variant detection and different types of WGS. But till then, we need to kind of uh, improvise. So if you're looking for short variations, there is no actual need for using long reads, so we go with short reads. But yeah, when you are trying to detect those big variations, it's optimal to go with long read data. And adequate read length that comes with the type of technology you're using for WGS, and also proper genome coverage. So like you would want to have a good coverage across the genome, because if you have like these ununiform regions where you have just one or two reads that are supporting your variant, it's very hard to like confidently claim that a variant is there. So yeah. Okay, so for processing reads after mapping, so what we do is, this is the basically the workflow. So we have this raw reads mapping when like using BWA or other types of alignment techniques. We have a we'll have a alignment file. um, After that, what we do is local alignment, local realignment. We'll talk about what it is in the next slides. After that, duplicate marking, then base quality score recalibration, and ultimately you have the process read. So this part of the pipeline is basically processing of the deeds post alignment. So before this, there will be the alignment. Before alignment, also there will be some sort of pre-processing, as we saw with fast queue. You will see what kind of reads are there, what are the different caveats of the data. You'll be able to see that. After that, you'll align the data, and after alignment, this is basically post alignment workflow. So you have the raw reads, then you do some sort of local alignment, you mark the duplicates, and you do some base quality score recalibration. This is important. Why we do this will while we'll do this, we'll talk about it later. But ultimately, what you generate is basically processed reads, and after this process, you can directly use a variant caller to basically check for different types of variants in your data. Okay. So let's start with a local realignment. Why is it necessary, and like why do we do it? So, some artifacts may arise due to the alignment stage, especially around insertion small indels. So these are, remember, we are talking about indels. When we talk about indels, it's mostly less than 50 base pairs. So there can be insertion deletion more than 50 base pair as well. We call them structural units. So when we are talking about indels, it's mostly less than 50 base pairs. So uh, some artifacts may arise due to the alignment stage, specifically around indels where reads covering the start or the end of an indel are often incorrectly mapped. This results in mismatches between the reference and the reads, misaligned regions, which can be easily mistaken for any signal. So that is why this realignment stage aims to correct these artifacts by transforming those regions with misalignment due to indels into reads with consensus indel for correct variant volume. Sorry. Yeah. So what happens is, around this in small indels, the reads are often misaligned because if there is a deletion, it's hard for the read to fully align there. So partially the read will align there, and rest of the read will not align. So things like these are can like what will happen is, so if you just imagine there is a reference genome, and there is a deletion inside your particular sample. So what will happen is, if this is the full read, it'll go and bind half like this part will bind, this part will not bind. So what can happen is you can easily mistake this part of the genome to be a single nucleotide variant. So if say one nucleotide is being covered by the gene, so the indel is around 20 base pairs, so the 20th base pair is being covered by a read, which is originating on the right of the genome. So a 20th base pair is covered. So the 20th base pair is not aligning, but the rest of the read is aligning. So it can happen that you're considering that as a SNP. The variant caller might consider that as an SNP. So it is very important to realign these misaligned regions, which and correctly kind of characterize them as indels. So what this does is transform these regions, small regions, with misalignment due to indels into reads with consensus indel for correct variant calling. So you basically you make a tag or use something basically characterize that part of the read as an indel compared to the rest of the read. So that when the variant calling is happening, you do not consider an SNP or something else for that particular region and not as a small index. So this step is actually very important. If you do not do this, there is high chances that you will get a lot of false positives, and even uh, like you can miss out on yeah, the different types of variants, which are basically false negatives. So it might be there is a 20 base pair indel there, what you can get is like 20 different SNPs in place of that. So it can very well happen. So it's very important to realign this part so that your variant caller does not confuse this with something else. So these are different realignment softwares that are used. So GATK Indel Realigner is a very widely used one. Others include Indel 2 and SRMA3. If you want, you can go and check out the documentations for this. But yeah, so these softwares does your local realignment. The inclusion of the realignment stage in variant calling pipeline depends on the variant caller used downstream. Actually, that is the caveat. So if your variant caller already has this integrated information to realign or to look for reads and not incorrectly claim them to be different types of variants when it is something else, if the algorithm is coded like that it does the realignment itself, then you do not need to do this step separately. The variant caller it will do it for yourself. But it's safer to do it because you don't never know what kind of variations might arise. So uh, this stage might be of value when using non-haplotype aware variant callers like GATK's unified genotype per 4. However, if the tool used for the variant calling is a haplotype aware variant caller like these callers, then it is not needed nor recommended. The GATK recommendations, starting from the 3.6 release onwards, and the guidelines for the functional equivalence of 8 also vote against this stage. So the newer callers that are coming out are mostly haplotype aware callers. So for example, haplotype caller um, from GATK, they already have this information encoded within them, so you do not specifically need to do this realignment. So in most newer pipelines, you'll see that they are skipping this local realignment uh, step because the variant caller itself has enough tools to identify these regions. So ultimately, the characteristics of the dataset at hand would dictate whether the realignment and other cleanup stages are needed or not. Uh, so there are certain papers which argue against this PCR duplicate removal, and there are some papers which recommend all stages of cleanup to be applied to dataset in hand. But there are conflicting reports on this. But depending on what software you are using and what check that documentation and what they're saying. So use your basically if like process your data in coherence of how your software actually expects it to be. So it is always better to look into the software and check if it requires, does not require, because it will always mention things like that. So yeah. Okay, so the tools that are used for this duplicate removal, sorting, and basically mostly sorting and duplicate removal and conversion of SAM to BAM. SAM is basically uh, the the SAM is basically the text file format, and BAM is the binary format of the same alignment file. So these conversions, these different types of uh, like what to say, different types of sorting of the BAM files and different types of duplicate marking are used done by the SAM tools software. You can easily download this. Sir has showed you. I'll still show you here. So this is the SAM tools homepage. You have the source release here. You can download it directly from here, or you can go to downloads here, and so you'll need SAM tools and BCF tools both. Uh, Both of them come with this HTS library, so you do not, if you're downloading both, you do not require this HTS library to be installed separately, but it's very simple actually. So you download this, and there's a uh, configure option or the configure executable inside the software. I'll just quickly show you. So I'll just log in a different window here. Yeah. So yeah. So these are your tar files. When you'll download them, and you just need to do a tar minus zxvf. So once you unzip this, these are the folders that are going to be created, and inside these folders, you'll find this configure file. So configure executable, you do a dot slash and you type configure and click enter, so it will configure all the scripts. After that, you have to make use the make file. So here's the make file is somewhere here. You just, oh okay, I have already made like compiled this, so the make file is not there. There will be a make file here, so you just do a make command and a make install command, and this whole thing will be installed, and you'll find this executable there. So this is the samtools executable. If you just put it here, it will give you all the options. So, yeah, it will allow all the options. So yeah, you can do a lot of stuff with samtools. We'll are mostly looking for samtools indexing. So we use FAIDX we use for indexing FASTA, FQIDX we use for indexing FASTQ files. Other than that, we use SAM tools for, as I said, like converting from SAM to BAM and vice versa, and also different types of sorting. And so this is the sort command. So we use it to sort alignment files. Uh, you can split a file by read group as well. So you can do a lot of stuff using SAM tools. You can check this out in the manual as well. They have written a lot about this. So this is something that is used for these different uh, parts of the pipeline. So what I'll do is I'll run these small commands during the end of this session, but yeah, bear with me. So now we come to the base quality score.
I have a query.
Yep.
Sorry if I miss out. We can use SAM tools as well as Picard, either of
Hmm.
the tools for the removal of
Yeah,
duplication,
yeah, yeah.
right?
You can use both. So SAM tools and Picard both. You can use.
Okay.
There are other softwares as well. So you'll have like a list of multiple softwares which does similar things. We are using SAM tools because it's the most popular one. Picard is also doable, but Picard uses Java, so you have to configure your Java inside your system well, so that you can use Picard. Not all the systems have Java, but SAM tools is easier to configured. So that's why I'm using SAM tools. No, no other
Okay.
reason. Yeah. Okay. Just a second. Uh, yeah. So now we come to the BQSR step, which is the base quality score recalibration. So it basically uses two. There are two steps. So I'm talking about BQSR in terms of the GATK BQSR tool. I'll just show you quickly. So there are other BQSR tools as well, uh, but I am talking in terms of this BQSR. So GATK module has a BQSR inbuilt in them. So you can go through this if you want to have a clearer idea, but I'll just leave it in the chat if you want to go through it. Okay. BQSR involves two steps in this case. One is the base recalibrator tool builds a model of covariation based on the input data and a known set of variants, producing a recalibration file. Then there is this apply BQSR command or the tool which adjusts the base quality scores on the data in the data based on the model, and it produces a new BAM file. So why is it required? The scores produced by the sequencing machine, the quality scores that we see, the qual scores, the Fed scales quality scores that are produced by the sequencing machines, they are subject to various sources of systematic, which are basically non-random technical error, leading to over or underestimation of base quality scores in the data. Some of these errors are due to the sorry. Some of these errors are due to the physics or the chemistry or how the sequencing reaction works, and some are probably due to manufacturing flaws in the equipment. So this PQSR process applies this machine learning model to remove these errors empirically. So for example, if we can identify for a given run where it has called 2A nucleotides in a row, the next base we called had a 1% higher rate of error. So any base call that comes after 2A in the read should have its quality score reduced by 1%, which is natural because you're systematically in your machine, you know that if there are two A's in a row, the third nucleotide, there will be a 1% higher chance of error. So any nucleotide that comes after two A's should have a lesser quality score, even compared to the quality score that is been uh, given by the sequencing machine. So you're correcting for those different errors that can possibly occur. So it is done over several covariates, mainly sequence context and the position in read or cycle, in a way that it is additive. So the same base may have quality score increased for one reason and decreased for another reason. So there can multiple processes go on for a single quality score. So for a particular error, it might go up. For a particular error, it might go down, and you can add these two errors depending on which one is greater. It'll go up or go down. So this allows us to get more accurate base qualities overall, which in turn improves the accuracy of the variant calls, because during your variant calling, different softwares will take into consideration the call value of your particular base call. So if that base quality score is not properly calibrated, what will happen is uh, you will get inflated confidence or you'll get deflated confidence on a particular variant around that region by the variant caller. So it is advisable to use BQSR before doing variant calls, so that your quality scores are all right and it has been corrected, so that you can have a better representation of the variants. Now we come to the another type of variations, which are known as the structural variations. As I was saying, anything greater than 50 base pairs. So there are two major types of this SVs. One is copy number variant, and one is copy neutral variant. So in copy number variant, a part of the genome is actually added or deleted. So the copy number increases or decreases. So that's why it is known as a copy number variation. This can include insertion, deletion, or duplication. Sorry. Yeah. Now, this can include insertion, deletion, or duplication. Like these are the three major things that can happen. And the other is a copy neutral variation, which primarily includes inversion. So a part of the genome has been inverted, or a part of the genome has been cut from one part and pasted in another part. So that's what a translocation is. So in these two cases, the amount of DNA does not change. So that's why it is a copy neutral variant. In terms of copy number variation, either it is increasing in case of insertion or duplication, or it is decreasing in case of deletion. So uh, the, the base caller, sorry, the variant callers in terms of structural variants, they classify these two differently because the the evidences that they look for a particular variant being a copy neutral or a copy number variant is the depth of that particular area of the genome. Say, for example, if there is a deletion in this part of, say, a big deletion has happened in this part of the region, so what will happen is you'll have no reads that are going and aligning to this part of the reference region, because in your sample this has been deleted out. So when you're checking the coverage of the entire genome, this part of the genome will have less coverage compared to the rest of the genome. So you can you can intuitively think that there might be a deletion because no reads are coming and aligning to that part of the region. So that part of the region is not present in your sample. On the other hand, if there is a duplication like this, so what will happen is, so all across a particular region, so say for example the middle region has been duplicated and the flanks of the duplication region are fine. Okay, so what will happen is the reads will come and align to the flanks. So the coverage around the flanks will be same, but in the duplicated region, the coverage will dramatically increase because you have duplication in your sample. So a more number of reads has been generated from that particular region of the genome compared to the other parts of the genome. So what we will see is the coverage getting suddenly increasing in, in this part of the genome compared to other flank compared to its flanks and other parts of the genome. So if there is a dramatic increase in the number of reads aligning to that part of the genome, you can intuitively think of it as a duplication. So these are strategies that are uh, adapted by these variant callers for big variants. So these are like big variants. We are talking about more than 50 base pairs. So, like in most cases, it will be uh, around 100. Mostly we'll find is around 100, but there will be some very big duplications or deletions. If you go and look into Y chromosome, you'll find like very big duplications, like 70,000, 80,000 base pair duplications. But there also you have to think about the case that Y chromosome has a lot of repeats, so it is very hard to claim that a duplication has taken place in a region of the genome where there is a lot of repeats. So there are all these caveats into place. You need to have enough evidence so that you can confidently say this part of the region has been duplicated or deleted or inserted or whatever, because it's a big chunk of DNA. It's not a small one. So yeah. So as I was saying, detecting SVs, depending on, like, say for example, you are trying to detect SVs using what to say paired end sequencing, for example, Illumina. So if there is an insertion here, so uh, in this is your sample. What happens is this is your sample. In your sample, you have these two opposing reads that has been generated, and during what to say Yeah. So during alignment of these reads, what you see is these reads are getting like closer to each other or far away from each other. So the distance between these two reads generated is already recorded when you are generating the reads in your sample. So if these reads are moving away from each other, that means there has been a deletion. If these reads are moving, if these reads are moving away from each other, that means there has been an insertion. If these reads are coming very close to each other, that means there has been a deletion. So also, if you also check the orientation of the reads, say these two reads are supposed to be oriented opposite wise, but if during alignment they are getting oriented in the same side, that means there has been some sort of inversion in that part of the genome. So this is discordant this is evidences from discordant read pairs. There can be also evidences from split reads as well. So a part of the reads align to one region of the genome, another part aligns to another region of the genome. So these kind of evidences also can tell you that there has been a deletion, or there has been a translocation of the particular region of the genome, because one part is aligning, say, to chromosome 15, another part is aligning to chromosome 1. So that means there has been some sort of translocation between these two chromosomes, so there is a structural variant here. So there are different types of evidences and algorithms uh, and variant calling software. So one of the major variant calling software that we use even is known as Manta. So it uses this split read um, technique, and also paired read, discordant paired read technique as well, both of them, to detect structural variants. So these are, like, what to say, for paired reads you can do this, but if you have long read data, this you do not need to go into so much caveat. Your long read data will most probably cover the whole structural variant if it is around like 100, 200, 500 base pair range. So you do not even need to think about these all stuff. So these techniques are mostly used in short read stuff. So Manta is a software that uses this kind of stuff, but in long read it's much more easier to use. So one of the long read calling softwares is known as Sniffles. They also use some part of these algorithms, but it's mostly not required because your read will cover the whole variant. So from alignment only, you know what kind of variant is there and how long the variant is. So now we've called variants. We've like we have uh, learned about how we call variants, what are the different uh, evidences we look for variant calling and stuff. Now, how do we store these variants in a file? So we have a standardized file format, which is known as the VCF file, uh, which stores this variant data, both SNP and SV data. So yeah, and uh, this type of file format is usually used in large-scale variant mapping projects such as IGSR. So IGSR is a project that did the thousand genome project. So here you can look about what the project did, and it's basically this is the thousand genome project as well. So how they did the project, and this is actually very interesting. So they took samples from all across the world, and firstly it started with 1000. It's I think this has reached to around 2.5 to 3,000 till now. What they try to do is collect different but collect data from different ancestral groups of the world and create a consortium of thousand of the samples and publish the data and kind of compare and check for SNPs, SVs, how different are we in terms of different variations in the genome and everything. This is actually a very sorry, this is actually a very interesting thing if you want to go through. And actually, you can use dummy, not dummy. These are not dummy. These are actually actual data. So you can use VCF files from this project. So there are places where you can download 1000 genome VCFs, and like these are human samples. You can run these samples in your pipeline and see variance, and you can compare the variance that they have found to what you have found. So you can have a kind of comparison report as well. So you can do a lot of stuff if you want with this data. So this data is available if you want to look into it. Okay, so yeah, so this VCF is a standardized format for storing the SNP or the SV data, variant data, and it is important in terms of it is standardized in the downstream processes as well. So after you have found the different type of variations, you would like to know what is the functional impact of these variations. So and how frequent are these variations. So what you'll have is further downstream annotation processes, which will basically pick up data from different kind of databases that include this thousand genome database. So this thousand genome database will have these SNPs, excuse me, these SNPs and SVs that they have already found, and they'll have a frequency associated with those particular SNPs and SVs for that, though, for its own cohort. So you would want to know how frequent is that variant, is the variant affecting, is the variant falling in any part of a gene? Is it causing any type of mutation that is affecting protein synthesis or not? Those kind of information we get from annotation tools. So we'll talk about annotation tools in the last slide. But the standard input for those annotation tools is also VCF. So VCF is something that is standardized across the whole pipelines. So this is like this is something that you will daily use if you are working in genomics or whatever. So VCF is the preferred format because it is unambiguous, scalable. Scalable is very important because the data size is very big. It needs to be scalable and flexible, allowing extra information to be added into the info field. So what happens is if you do annotation and after annotation you ask it for a VCF output as well. So you give the input as VCF and ask for the VCF output as well. So in the extra information from the annotation will be stored in the info field. So it can directly store annotations and stuff. You do not need to do anything else. So basically, these are tablet delimited files. You can read these files in basically if you are using Python, you can read these files in pandas data frame as well because these are tab separated files only. So I'll show you what a VC file looks like. But before that, this is actually the main part of the VCA file. There will be a list of headers before this part comes along, but this is actually the main part, and this is how the variants are stored. So this is first you'll have the chromosome, then you'll have the position in the chromosome. If there is an ID associated with it, so here it is given as an RSID. So RSIDs are like basically SNP IDs that are given from DBSNP. If you want to take a look, so DBSNP is a very good tool. So it tells you about different. So I have already searched for this RSID. So it will have an RSID associated with a particular variation. So these are the two alleles that are present here. You go into the variant details, you can find the different alleles, and you can find what kind of variant is it is, which is mostly a missense variant with an amino acid change and stuff. So the DVSNP database gives you all this information for different types of variants. These are all kind of uh, stored in this database. You also can look into clinical significances. So one of the clinical significances might be a pathogenic variant which can cause a particular disease. So these are all mm, types of information that you can get with the help of this ID. So this ID is basically this coding for this particular SNP. So so this particular one is a so I was looking into it because this is a rare variant that is found in the Polg gene. So Polg is a gene that is involved in different neurodevelopmental processes. So if for this variant, uh, if this variant is present, there are severe, what to say, severe abnormalities in neurodevelopmental disorders leading to several type of disorders like this. So this kind of information for a particular variant, what is the functional impact, what kind of disease is it implicated in, these kind of things you have to go to databases like DBSNP to find out. So there are other databases as well. One of them is ClinVar, which will tell you if there are any clinical significances to this particular variant. So it tells you like it's likely pathogenic. So yeah, this is highly possible that if this particular variant is present, the person will develop that particular disease. So there are different types of these databases that are present. So you want to collect all this information and annotate your SVs or SNPs so that you can have an idea about, you can prioritize your variants in terms of like which is the most pathogenic, which is the most important one, which is the one causing disease, which is the one that is benign, stuff like that. Okay. Uh, yeah. So as I was talking about, this is the format. So if you have an ID of RSID associated with it, you have it here. The ref is the allele that is present in the reference. So it is not always the major allele; it's just the reference allele. And the alternate is basically the SNP that is there. So a T to a change has been done in your sample. So this is what it means. So if you write it in one format is will be 11, then you put a and then you put a colon, then you put the position, then you put another colon, you put the reference, and then you put another colon and put the alternate. So this is also one way of representing these SNPs. So yeah, and qual is telling you the quality score of this particular variant. This is, remember, this is the quality score of the variant, not the quality score of the base. So during variant calling, it will also uh, assign a quality score to the variant. So, and another filter is basically a pass filter. So this pass filter, what it is telling you, it has passed certain criteria. So it has a qual score greater than this, it has a genotyping coefficient greater than this, or it has some. So you have to define a set of parameters which will tell you that if it is a pass or a non-pass variant. So if those particular uh, things are present, it will be a pass variant. If those, basically, if there is enough evidence, it will be a pass variant. If there is not enough evidence, it will be a non-pass or a low depth or a something else type of variant. And in the info column, you have all this information. So what this means, AC is the allele count. So there is, the allele count is 1, which means that uh, only one individual has this particular allele inside the whole cohort. Then there are these AF. AF is basically the allele frequency. So what is the frequency of this allele? And now are different uh, population allele frequencies. So African allele frequency is this, American allele frequency is this, East Asian allele frequency is this, European allele frequency is this, and so on. SAS is basically our population, the South Asian population, the Indian, Pakistan, and the Bangladesh, Indian subcontinent population. So SAS population allele frequency is this. So these kind of different kind of information can be included in the info column, and in your format it will be the genotype present. The genotype is given by this type of like notation. So what a 01 means, it's a heterozygous variant. There's one strand has the variant, another one does not have. Now one thing you notice, this slash between sorry, take the pointer here. Yeah, this slash between 0 and 1. So this can be either a standing slash or a slanting slash like that. So if it is a pipe, a standing slash like this means this is phased. So what does phasing means? Phasing means to check if the variant is coming from the father side of the allele or the mother side of the chromosome. Sorry. So there, in one chromosome there will be two strands. One strand will be inherited from your father, one strand will be inherited from your mother. So this phasing tells you exactly on which strand is the particular variant present. So this is present in the second strand. So if there is phasing, you'll have this straight pipe symbol. If there is no phasing, so you do not know which variant, which strand it is in there, but you know it is present in one strand, but it is not present in another. So you'll have this slanting type of slash 0 1 or 1 0. So in case of phased genotype, 1 0 and 0 1 are different. But in case of unphased genotype, 1 0 and 0 1 are the same, which is telling you it's a heterozygous variant. So the possibilities for an unphased genotype are 0 0, 1 0, or 1 1. So either it is homozygous present, either it is heterozygous present, or it is homozygous not present. So these are the possibilities. But in case of phasing, you also know that information, if from which strand is the variant coming from. And this header is telling you the sample ID. So this is basically the sample ID that I was showing here. Here, this is the sample ID. So yeah. So what will happen is, if you have a cohort data, so you have like SNPs from 1,000 individuals, what you want to do is have one VC file, and it, in that file it will tell you the variants and in which sample the variants are present in. So from here to here it will be the same, and after that there will be different columns for different samples, and inside each column for different samples you'll have the genotype for that particular sample. If it is not present, it will be just a 00. So this kind of information can also be added into a VC file. So you can merge different VC files together and get one VC file which will tell you uh, all the genotypes of the variants for all the samples in one VC file. So like that. Now inside the VC file, you'll have firstly the meta information lines, which are the header lines, and then you'll have the columns like this. So before these columns, you'll have the meta information line. So it starts with these like double hash values. So first, in the first line, you will find something like this hash file format, which will tell you either it's VCF, this is the VCF file format version 4.0. The current version is I think 4.2, but yeah, it'll tell you the version. And what it also tells you are different like different elements uh, of the VCF file. So I'll just directly show you the VCF file. That will be easier, I think. So I have a VCF file here. Yeah, yeah. So these is your basically your header lines. From here, your basically the format starts. But before that, these are all your headers. So this basically tells you the information inside the VCF file that is coded. So first, you have the file format. Then it tells you what does the pass mean. If there is a pass in the basically in the VCF, what does the pass of the ID of the particular sample, particular SNP means that it has passed all filters. Then you have the commands that you have used. So I have used this particular command. It's there. The reference file it tells you. Then it tells you the config IDs. So these are mostly these are chromosome IDs that are like chromosome numbers that are coded in IDs, but in like most cases you'll find this chromosome 1, 2, 3, 4 like that only. So these are different chromosomes that are there. Then you have the alternate allele. Then you have what does an indel mean? Indicate like what does a tag of indel mean? So it means that there is an indel. Then does the tag of IDV mean? So these are different types of it. What it tells you is basically what is the encoding of different of these short forms that are used. What is a GT? See, what is a GT? GT is a genotype. GT means a genotype. Now what is a say for example, what is an AC? So allele count. Allele counts in genotypes for each alternate allele in the same order as listed. So it basically explains you the different things that have been used, different notations that has been used in the VCF file, and it gives you a description of what that particular notation means. So that is all that you have in a VCF file in terms of the header. So I'll not dwell on this much because it's not much interesting, but you'll have all the information that you need, like that you require to understand the VC file inside the header, the meta information line as well. And this is the basically your VC file. This is how it will look like. So these are all the columns that I was showing here. It's all there: chromosome position, ID, default, qual, filter, info, format, and everything. Okay. So this is basically the name of the sample. So I've given it as the whole path, so that is why it is taking like that, but this is the name of the sample. So Chrome will tell your chromosome, position will tell the coordinate of the variant, ID is if there is an ID identifier associated with it, it will be there, otherwise it will not be there. For the reference allele, it's basically the reference genome, what is present in that position. Alternate is basically your SNP, what is present in your sample. Quality score is the qual score. Filter, if it is a pass or a fail, it will tell you. And info will tell you further information like allele count, different types of allele frequencies, and everything. In the format column, you'll have the genotype, which will tell the genotypes of the particular samples for the particular SNP. And in the basically your sample column in your sample column heading, it'll the previous column told us to expect to see genotypes here. The genotype is in the form of 0 type 1, where 0 indicates the reference allele and 1 indicates the alternate allele, that is, it is a heterozygous variant. The vertical type indicates that the genotype has been phased, as I was telling, and is used to indicate which chromosome the alleles are on. If the slash is a slanting one rather than the vertical pipe, it means we don't know which chromosome they are on. So, yeah, this is how the EFI looks like, your VCF file. Now, in the last part of this session, we'll discuss about variant annotation. So we looked at what are variants, How do variants, how are these variants detected? How are these variants saved in a file, the file format of VSIF, and now what are the annotation strategies? So, what annotation do you exactly want from a variant is a thing of concern. So, basically, either you'd want some sort of like what if it is a functional important variant or is it something else? Just give me a second. I'll show you an example. Just I'm just stopping the sharing of my screen for a moment. I think you can see my screen. Yeah. So this is how an annotated file looks like. So you have chromosome position ID default. Now you have an annotation. So what kind of annotations can we have? So let me filter this. So you can have these kind of tags. Is it a downstream element? It's if it's an exonic, intergenic. So different types of softwares have different types of categories. This is a ANOVA annotation. So we have these categories. Also, along with that, we have ClinVar categories. Is it what kind of things can it affect? Is it a benign variant? Is it a drug response variant? Is it a risk factor variant or something? Also, you have these are like frequency of nomad uh, for that particular variant. What is the frequency of that particular variant in the nomad cohort? What is the frequency of that particular variant in other cohorts as well? This is the 1,000 genome cohort. So, and also you have associated different type of scores associated with it. So this CAD, polygen, these are scores that calculate risks from this particular SNPs, and they give you a score telling you how probable is that, but like how dangerous is that particular variant. Also, along with that, you have stuff like amino acid changes. So, what kind of amino acid change is happening and in which gene it is happening. Okay. And you have other type of things, so like what are the different type of frequencies in different type of populations, and also most importantly what are the genes associated with this particular variant, meaning like in which gene does this particular variant fall, and if it is falling in an exonic region, is it a synonymous variant, it's a non-synonymous variant, it's a frame shift variant, what type of variant is it? So this type of information is what you get in annotation. So after annotation, so uh, yeah, so ANOVA is a tool that does variant annotation, and it does annotation in different regions. So it does gene-based annotation, relationship to the gene, if it is an exonic, intronic, intergenic, as I was showing you, if it's like if exonic, if it's a synonymous or a frame shift or a non-synonymous, what exactly it is, exact amino acid change if it is exonic as well, and in the regions, like if it is a part of microRNA region or if it is a transcription binding site, if it's a conserved element or something, and also filters like different type of allele frequencies reported by say a 1,000 genome project, prediction scores compiled in DBSNP. So what it does is like pulls out all the information available in different databases for a particular variant, and it annotates your VC file with it. So uh, that is all in terms of variant calling and basically VC files and annotation. Uh, if you don't have any questions, we can move into the assignment part.
I have a question.
Yeah,
I read it somewhere. I don't know. I might be wrong. That BQSR step, base quality recalibration step, is quite computationally expensive
yeah, yeah,
step. So. What if we skip? Matlab, is it possible if we? Matlab,
yeah, yeah.
uh, can we skip that step?
Yeah. So the thing is, it depends on your data and the variant caller that you are using. So if your variant caller supports non-BQSR data, It's well and good. So what happens is, if you do BQSR, the probability of a variant having a particular confidence score, or a probability, or the call score that is given to the variant, if you do BQSR, the call score might increase if you have good enough evidence, and the call score will decrease if you do not have good enough evidence. So what it does, it like kind of secludes the two, if it is a confident variant, if it is a non-confident variant. So it kind of makes a gap between these two types of variants. If you do not use that, the thing that will happen is, if you you might get similar scores for variant that is a like that is high confident. Also you might get a similar score for a variant that is not so high confident. So the differences between the call scores might be a problem. But if you are considering, like, if what happens is, maximum cases those will be pass variants only. So this BQSR step will affect only a subset of variants that are kind of ambiguous in nature. So in most cases you can get away with without doing BQSR, but for best practices, so in GATK only, there is a best practices page where it tells you what are the best practices for WGS pipeline usage. So there also they tell, okay, it's better to have GQSR, BQSR than not to do it. But so in case of exome sequencing, for example, I do not use BQS. I use the variant caller, because I'm not much bothered about BQSR, because BQSR only will help in mostly in the interonic and the intergenic region parts. If there is an exonic variant, there will be enough support for it anyway. So yeah, so depending on your usage, I'll not say do not use it, but in some cases you might get away with not using.
Okay. So I use one more thing. So I usually use iWar for VCF. So, matlab, any comments about that tool? iWar, about using iWar.
I'm not sure
iWar.
what is the spell.
iWar.
I war.
iWar.
Okay.
VIVAR. That's it. iWar. VAR.
Okay. Okay. I've not used this ever, but okay.
Okay.
I have to check and tell you.
Because I
I've
was
not
not
used
sure.
this. Uh,
Okay. Okay.
this is a this is used for viral amplicon based sequencing mostly.
Right, because I usually work on viruses,
Okay.
virus
Okay.
genomes.
Okay.
So,
Okay. So I have not used this before, because I work on human data mostly. Okay.
okay.
Okay. I'll take a look. I'll take a look. I'm not sure otherwise.
Okay, okay, okay. Fine.
Okay. So let's go to the assignments then. Actually, let's try these commands out. Samples. So here are my different files. So this is I have downloaded from the folder that sir has only shared. I guess you have the link. So let's take out the SAM file. This is the SAM file, and let's try to convert it into. So 1 second. Okay. So you have SAM tools here. So these are the different commands. So what we'll use is SAM tools view. So this is the one that gives you the conversion, SAM to BAM, BAM to SAM, and CRAM as well. So the command will be like something like this. Oh, okay. Just let me first copy the file path. This is your SAM file. Okay. So what we do is uh, SAM tools view. So this is the view. We have to give 1 second. Okay. Let's just look at this the options for SAMtools view. So these are like SAMtools different types of commands. Inside those you can give different types of inputs as well. So this is the SAMtools view help command. So in this, so we want to have an output of a BAM. So will give this in output. We'll have to have a is this thing? Yeah. So we'll use the auto detected format of the input. We'll not give the input format directly, and we'll use the view file and convert it to BAM. So this is SAM tools. We use view. We use two tags. One is the output BAM and is the like auto detected input. Right? So minus B capital S. This is your SAM file. I have put the whole path here because the path is somewhere else. So, and I'll want to convert it to a BAM. So, put output test1 dot BAM. So, SAMtools view, I use like this B tag for output BAM, S for auto detecting the SAM file, and we'll convert it to a BAM. Okay, so it's done. Let's check this one. See, the output BAM has been created here. Okay, so the answer to this is actually SAMtools view. SAMtools view, then you give a tag that you want the output as a BAM, and then you convert it to a BAM file. Right. So one thing to notice, it does not directly give you the BAM file like that. So you have to put a pipe as a output and then put the BAM file like this. Okay. Okay. Yeah. So the answer is all the first three. So except for base recalibration, during duplicate marking of the SAM tools, it sorts the read by name, not by coordinates. Remember, because like after this marking duplicates and everything, and before just variant calling, you need to sort the BAM file by its coordinates and everything. So it does not sort by the coordinate. It fills in mate coordinates and insert site fields, and it also sorts on base of chromosome number and coordinates, right? So this is the process here, pipeline. Yeah. So Mark duplicates, will remove duplicates, print some basic stats. Before that, you need to sort with the chromosome number and coordinates. Before that, if you use fixed mate, this will fill the mate coordinates. This is also optional. You can use, cannot use. And before that is basically sort the alignment based on the name of reads. So if you're doing removing duplicates, it does actually, you have to follow all these four commands. So first you need to uh, uh, like what to say, sort by the name of reads. Then you need to fill in mate coordinates, which is optional. Then you need to sort it in base in terms of chromosome number and coordinates, and then it'll remove the duplicates. Even if you do not remove the duplicates, you can directly use a variant calling software on this particular file as well. So if you are not interested in removing duplicates. So BQSR actually uh, does everything except for the identification of duplicate reads that is done by the mark duplicate SAM tool command. It allows us to get more accurate base qualities and improves accuracy of variant call as we discussed. BQSR can help improving variant calling or genotyping analysis. It's a measure of confidence in the base call, and it's the process uses known variants to mask out bases at real sites of real variation to avoid counting them as errors. So in the BQSR command, you need to give actually a VCF file that has that is a like a pre-given VCF file which has all the known variants. So it masks out those particular regions and computes the BQSR for the rest of the data. So BCF like BAM is basically the binary format for BCF. It is designed to store data and basically it's exactly VCF but in a binary format and it's smaller file size. It's not a text-based format because it's a binary format. So yeah. And it contains both variant and genotype information just like a VCF, and it's faster to process and manipulate because it's a smaller file size than VCF, and it's like what to say, it's encoded in binary, so it's easier to process. Is anybody giving answers? I'm not sure. Okay. Oh, sorry, I did not check your answer. Okay. Uh, okay. Uh, I am a little bit conflicted on this. So, seven, I think it's both like one and four. Heterozygous is basically the genotype will tell you, and also this thing. Both are genotypes only. So, a little bit conflicting. I'm conflicted. I'm on this one, but okay, let's go over it. So, allele frequency is given in the info column, which is correct. Total depth also in the info column. Everything, these are extra information, so these are all in the info column. The read depth will be in the format column. In the format column, it's okay, all right. Uh, the Q10, which is the quality value, will be in the second and the sixth column. So sixth in quality is there. In the filter column also it can be present. So this is also all right. Five is the haplotype quality is basically present in the format column. So format will have different types of quality, which includes normal variant quality and the haplotype quality as well. And mm, phasing is given by in the genotype. Okay, so phasing and genotyping is okay. But the seventh one, I think it should be both one and six. Sorry, one and four. This thing, the actually the eighth options last uh, option should be here as well. So I that is my thinking. I'm not sure the answer is given like this, but it's okay. But heterozygous will be given by the genotype and what is the phased and the unphased genotype given there. So yeah. Uh, so the S50, what it actually means is like it is present in less than 50% of the samples. So uh, like it's a tag that tells you not exactly about the allele frequency. Allele frequency will be given in a separate way, but it tells you how many samples actually have that particular variant. So yeah, if the variant is covered then less than 50%, will have this S50 value, which means like this is not a very common variant. This is might be a relatively common variant, but it's not a very common. It's not present in 50% of the samples. Okay, yeah, it's correct. So frame shift will occur when the amino acid frames shift actually. So there's a codon, so AUG and like say CTG or whatever the codon is, the CUG. So T is not there in RNA. Sorry. So these three codons actually will code for one amino acid. So if you shift the mutation in such way that the three codons actually the combination changes. So from one to three, it becomes two, three, four. So that it causes the frame shift mutation. So insertion and deletion can cause this mutation, and also it can result in the production of truncated protein. They are not always silent mutations. Actually, most times they are not silent mutations. If a frame shift is silent mutation, it's very rare. Yeah. So, okay. So the answer is E. It does not have any protein information. So what it'll have is it's mostly gene information. What kind of variation it is? Is it a like kind of a functional impact? Is it in exon or is it in a contiguous sequence which begins with nucleotides and a start codon and ends with a stop codon and something like this? So it'll have all this information, but it'll not have information about the protein. So that's not in this particular type of format. If you don't to do that annotation, you need to form like a bed file or a CSV file like that. Okay. So in GTF files, you have the hierarchical organization of features, and I'll show you the GTF file once. Uh, so you can look into this. I have not covered this, so I'm not going to detail. So this page actually gives a very good description. This is the ensemble page about DTF file format. So please check it out if you have any issues. Actually, you can use GTF, GFF, except FASTA and VCF. You can use any bed file is actually a very interesting format to use for different annotations. You'll find different annotation databases having bed file formats. So, except for FASTA and VCF, you can use any of these file formats. GTF and GFF are actually annotation file formats only. Bed is something that is used. Bed is a very simple file format. So you have a chromosome, position, and then base. Instead of position, you have a chromosome, then start and end, and then you have the annotation. So it's a very simple file format separated by tab. So it's actually also very good. So instead of VCF and FASTA, you can use anything. Okay. Info will have additional information about the variant, allele frequencies, and everything I showed you in the example. The format is used for the data type and if GT, it will be written as like GT for the genotype, and genotype will be given there as well. And uh, the info and format field will use a key identifier. Actually, this identifier that I was talking about here, let's just let me go back. Yeah, so in format has this GT identifier. Info will have these ACA, these kind of small notations as identifiers. So uh, both will use these identifiers. So yeah, instead of the incorrect option here is the info field can store only a single key value, which is not the case. You can store multiple different keys, different types of frequencies, different types of other information as well. You can store. So yeah, the options are A, B, and Uh, actually, changes in protein folding, it's very hard to uh, point out. Like changes in protein folding will only occur when the type of amino acid has been changed. So synonymous mutation will not change the protein folding type because an acidic amino acid will be replaced by an acidic amino acid, right? But if a particular, say a basic amino acid has been replaced by an acidic amino acid, then the hydrogen bond forming and everything will change, then the protein folding will change. So synonymous mutation will not change protein folding, but still it's like hard to confidently say that it won't, but most cases it won't. And non-synonymous changes not only the DNA sequence but also the amino acid. So in that case, it might have protein folding implications in that. But claiming anything about protein folding is very hard. So yeah. Okay, so the first statement, germline variants can be inherited from either of the parents, while somatic variants are acquired during individual's lifetime. This is actually true. So germline variants are propagated through reproduction, and somatic variants are uh, generated through different types of environmental effects and other stuff. It's not that somatic variants are always pathogenic. There can be like they are benign variants, like there might be variations, but those are not actually affecting anything. So while highly be possible, and germline variants are also like not always benign, because germline variants can have those pathogenic variants which can cause those rare disorders, neurological and other types of disorders as well. And somatic variants cannot be passed into offspring because your germline is not mutated. Uh, Actually, it's the opposite. Germline can be passed through offsprings, and somatic variants cannot. And phasing haplotypes can detect parental contribution for germline mutations, as I was talking about phasing. So there are two strands. If you correctly identify which strand has been mutated, you can identify like if it is coming from the father or if it is coming from the mother. So that you can do. So the correct options here are A and D. So yeah, that's all for today. If you have any questions, you can ask, or else we'll close the session. Okay, then. Thank you, guys. Thank you for joining. We'll meet next week, uh, same time, 5 p.m. Okay. Thank you.